So a quick one-page review of DNA and protein synthesis. We completed this in class on Friday, the 22nd of March. So question number one, quick completion of the matching side to the DNA strand means that we're just apple in a tree, car in a garage. So here is the answer key to that. Below the G is a C, below the T is an A, below the C is a G. And then we continue on TCT, GGC, and TTA. Again, the only skill required to know this is that DNA is made of A, T, G, and C, and that A's and T's go together and C's and G's go together. Then we move on to step 2, in which we are going to create a protein. The original DNA strand is unzipped and it's present at the top there, TTC, CGA. You're going to make a messenger RNA strand using the same pairing rules that you did above, except RNA does not contain a T. It only contains U's or uracils. So if apples go with a tree, our first letters are A, and then under the T we get an A, and then under the C we get a G. We continue on G with a C, C with a G, and then a U under the A, because instead of a T, it uses a U. And then we continue on with CCG, AUA, CGG, and AUC. And the transfer RNA strand is going to dock up with the messenger RNA strand using, again, the same pairing rules, A with U, or T, and G with C. So under the A we get a U, and under the A we get a U, and under the G we get a C. And from there we get CGA, GGC, UAU, GCC, and UAG. Again, just pairing A with T, G with C, and with RNA, A with U and G with C. Now the amino acid is gotten from the chart below, and that's a messenger RNA codon sheet. And that's why I drew an arrow from the messenger RNA line down to the chart below, because we use the messenger RNA 3-letter codons in the chart to find the amino acid. So I'm going to look for AAG. And I find it by going down to the A row, across to the A row, and look in that box and get a G, which is the LYS, or lysine. So if my first amino acid is LYS, and then I look for GCU, and I find it by going down to G and then over to U in the first column, and we get alanine. And then I continue that process, finding GGC, and I get pro, and then ILE, and then ARG and ILE. Okay, and that is the protein strand that would be made from that original strand of DNA. Some things to notice. Notice how the DNA and the tRNA lines look almost identical, except the T's have been converted to U's. Because the great thing about this system is that if you have an original message and you code it in RNA and then you decode it, you get the same original message. And that's why the message can be conserved without errors most of the time. Works great. So there's a quick review of protein DNA, protein synthesis and DNA. And about time to have a quiz on it, don't you think?